Okay, today we will be looking at the brain, going over some basic neuroanatomy, and then demonstrating how to properly do some brain cutting when you get these in autopsy. Every place will be slightly different, but at least if you have a basic understanding of what you need to look for and why, you'll be better equipped to handle brain cutting and autopsy dissection. So you guys will go in more detail about neuroanatomy when you get into neuroscience, but that won't stop us from going over and appreciating some of the basic structures of the brain. When you first look at the brain, you notice it's got that characteristic kind of folded in on itself appearance. You guys learned that word as cerebriform, taken kind of from the cerebrum of the brain. You got your two main components being the cerebrum and the cerebellum that you can usually easily identify. The cerebrum we divide into several lobes that you should be able to identify. Namely, we have the frontal lobe up front, which is kind of where your personality lies. You have your occipital lobe in the back, which is primarily involved in vision. You have your parietal lobe, kind of in the midsection, which is deals with proprioception, kind of your awareness, and then your temporal lobe, which is sort of your feelings of being awake and just with it. So you should be able to look at the brain and identify those. When we do look at some of the gyri and sulcuses on the brain, there are a few that you'll learn in more detail when you get to neuroscience, but a couple of ones that you can identify particularly is the one between the parietal lobe and the temporal lobe here. That makes that kind of characteristic fold in the brain here. That is your lateral sulcus. So that one should be easier to identify. And one that's kind of tricky but is usually prominent on every brain is the central sulcus. And what you're going to want to do is look for the midpoint of the brain for the central sulcus. It'll be the dividing point between the parietal lobe and the frontal lobe. Next to that, you got your precentral and postcentral gyrus, and those have various functions about controlling your motor and your sensory receptions for the entire body. So those are kind of important in neuroanatomy. Again, looking at the rest of the brain, so we already mentioned that the cerebellum is down here, and we got our start of the spinal cord. This marks our brainstem region here, and the other prominent feature that you'll notice on the brain is this bulge on the anterior aspect of the cerebellum. That's your pons, and just below that will be the medulla oblongata. This brain, you can see some of the circle of Willis. We'll go over that in more detail shortly on a better specimen, and we don't have all of our cranial nerves attached, but you should remember those from anatomy. They don't play as significant role in autopsy dissection, but they'll actually be important for studying for your certification exam. So make sure you're at least aware of where those are. When we look at the brain, a couple of other prominent features that we'll notice is our optic nerves coming out of our optic chiasm. We do actually have our olfactory bulbs that are involved in sense of smell. And one of the structures we'll want to look for just behind our our optic nerve is our mammillary bodies, and this one doesn't have prominent mammillary bodies, but we'll come back to those and why they're important when we start doing our sectioning, and I'll show you those on hopefully a better specimen as well. But that's kind of the basics of the external anatomy. Now we're going to go over and look at the internal anatomy of the brain as well before we start actually doing our cutting. So now we're looking at a hemisection of the brain, and you should be able to easily split the brain into sagittal sections and see what I'm showing you here. Ideally, you'll also be able to identify part of the central sulcus we looked at earlier on the mid portion. It kind of stops halfway here. Another prominent feature you'll see on the midsection is this corpus callosum here, which is at the top of your lateral ventricles. Now, the ventricles are the space that contain all your cerebrospinal fluid. You should be aware of the different ventricles that we have. So we have our lateral ventricles on the left and right hemisphere of the brain, and that'll drain into your third ventricle, which is kind of the space right inside your thalamus. There's the cerebral aqueduct, which drains the third ventricle into the fourth ventricle, which this is a space right behind your cerebellum, or I should say anterior to your cerebellum. The thalamus is the central region, kind of the control center of the brain. The hypothalamus, which is a little disrupted here, is going to be sort of the triangular portion anterior to the thalamus. And extending from that, you would actually be able to see the pituitary, which we don't have in this structure, and it's actually kind of difficult to maintain on a cadaver brain after fixation to get the pituitary out. Usually get that a lot more in fresh specimens. In the back here, you should be able to see the pineal gland, kind of the posterior aspect of your thalamus. We can get a better glance at our midbrain, kind of the start of our brainstem. We see our pons structure here, followed by our medulla down here. And then on top of your thalamus, you actually have the fornix as well. So hopefully you can appreciate, again, the various lobes. We have frontal lobe, parietal lobe, and occipital lobe. Now, keep in mind that we do not see the temporal lobe, except for a little tiny component kind of hanging in the background there, but the other lobes we should be able to see without issue. Okay, so let's take a look at the vascular supply. Okay, so this specimen has the vasculature of the brain in much better detail than the one we were previously looking at. The one thing we are missing is the start of the circle, which is going to be the two posterior vertebral arteries. Remember, those are coming off the subclavian, and those are going to follow up from the spinal cord, and they'll merge to form this vessel here, which is your basilar artery. Right? From the basilar artery, you'll get the start of your circle. We have one vessel that's going to go all the way towards the anterior aspect and meet up with our internal carotid and middle cerebral. And that's going to be our posterior communicating. So that should be this one here, if we can make sure we find that. And that's not to be confused with the posterior cerebral. Now, the difference between the two is your posterior cerebral is going to be going up towards the posterior near the occipital lobe. And so you want to make sure that you can find, let's see if we can find it on this one here. So that actually could be our posterior going posteriorly, and then we have maybe what looks like our small posterior communicating going towards the middle. Now, most brains you should be able to see a cut internal carotid, which we see here. And let me zoom in a little bit so you can appreciate these structures. So the cut portion coming pointing inferiorly, that's your internal carotid. And that's going to be the anterior supply of the circle of Willis. And coming almost immediately from that, you should be able to see the middle cerebral here. Additionally, just off the internal carotid, and I don't know if we have a good example of it here, but you'll also be able to find your ophthalmic. And it's not really easily seen on a lot of brains, depending on how well you're resected, but that'll be going up towards the anterior portion. So we have our internal carotid, middle cerebral going up between that lateral sulcus, supplying the parietal lobe. Then we're going to get an anterior cerebral coming off of that, which we see here. So you'll see kind of a nice little union of vessels in the center here. So your anterior cerebral is going to come up and supply the frontal lobe. Now the anterior communicating vessel is actually going to be the one that connects your two anterior cerebrals. So it's a very small vessel, and it just joins both of those. So notice the difference between that and when we're trying to look for the posterior cerebral, which is kind of a nice longer vessel connecting the two, the middle cerebral and the posterior cerebral. The anterior communicating vessel is the only one connecting the anterior cerebrals. And that's pretty much your circle of Willis. This tends to be a common location for a lot of your Berry aneurysms. And when we're thinking about stroke patients, because of the location of the middle cerebral, if you think about a vessel or a clot of some sort, an emboli or thrombus forming and then breaking off, because of the location of the middle cerebral, just at the junction of the internal carotid, you can see that this makes sense that this would be the most common site for an infarct. So any sort of emboli that flows through the brain tends to find itself in the middle cerebral artery. And what that generally supplies is this parietal lobe here. And if you remember, we talked about how most of our motor and sensory functions exist in the parietal lobe, and particularly that pre and post central gyrus. Well, if we get an infarct there, that's when we start manifesting symptoms of the stroke. They could be sort of defects in our sensory or defects in our motor skills. And more particularly, we have what's known as Broca's area and Wernicke's area in the parietal lobe in this general region, which are manifesting speech. So Broca's is for speech formation, Wernicke's for speech understanding. Broca's areas tends to be most affected in stroke patients, and that's why you get that characteristic difficulty in speaking for a lot of stroke patients, because this region in the parietal lobe becomes infarcted and the tissue dies. So that's kind of an overview of your circle of Willis. And then you can also refer to diagrams to kind of get a nice picturesque view of that as well, and also some of your notes from anatomy. Now we're going to start looking at cutting, and I'm not going to cut this brain because it has a better vasculature supply, but I do want to show you what I was trying to show you in the previous brain is the mammillary bodies. These are two bodies that are just anterior to the brainstem here. So once you find your midbrain, just go anterior to that. You'll see these two little nubbins that are your mammillary bodies. Those are going to be your central point for cutting the brain. You want to find those, and we're going to make our first cut through those particular structures. So ideally, you're just going to go anterior to your midbrain. So I'm going to get my other brain, and we'll start doing some cuts. Okay, so after looking at some basic anatomy, now we are ready to cut the brain. Now the nice thing is there's not really a wrong way to cut the brain, and other places you go to may have their own unique approach. But there is a way if we're going to look for some basic sections, or if they don't care, if it's just a routine autopsy. There's some sections that we can look for various effects. And the first thing we're going to want to do, while you can cut everything as a whole brain cut, keeping the cerebellum and brainstem attached, I find it's often easier just to try to cut that away as best you can. And that way you'll be able to see your substantia nigra, which is an important section you want to take. Okay, so using just a regular scalpel, what I'm going to try to do is make like a deep cut just above the midbrain and try to resect the midbrain from the cerebrum. Alright, there we go. Now, the thing that should stand out to you if you do this section correctly is this brown discoloration, which is a very characteristic pattern in the substantia nigra. Now, when you take your sections, and you'll also be able to see this on your brainstem typically, we want to take a section of substantia nigra in order to rule out things like Parkinson's disease. In Parkinson's disease, we'll see actually a discoloration of this. It'll look a little pale. You might not see any brown discoloration at all. The brown is actually due to dopamine generation. So we see it looks good. I could go ahead and take my section here, which I might just do just to be done with it. And I already got this other one over here ready to go. So let's take a nice section. We'll save that for later. Now overall, there's 5 important sections that we're going to be taking, and that's one of them. All right, so the next step is I showed you those mammillary bodies on the cervical Willis brain. Now with the brainstem out of the way, we can see them right here. So we're going to use that as our cutting point. So go ahead and take your long blade, line it up with the mammillary bodies, and make a nice smooth cut through the brain. Now we haven't looked at the frontal cuts yet, but here is pretty much a lot of your cerebrum sections that you're going to want to be taking. So a couple of things we should note that we've already looked at the lateral ventricles here. We can see our thalamus in the center there. Some other structures you should be able to identify, and again you'll go into more details about the purpose, is this basal ganglia, which is these brown regions on the central component of the brain. So you have your caudate nucleus up here, this brown nucleus, just lateral to your lateral ventricles. You have your globus pallidus, these little brown regions kind of in the center here, making this little triangle, and the outer side is the putamen nucleus. Additionally, we have this little curly Q here. This is going to be our hippocampus, and this region above is going to be the amygdala, which is kind of your sort of your reptilian brain. The hippocampus is important, and the way to find that is like kind of like a cinnamon roll, or Roseanne likes to call it like a little captain's hat. You just kind of look for this little swirly swirl. You want to take a section of that, and that's really important for demonstrating signs of early hypoxia that may not have been noticed clinically. So typically what we'll do is we'll get a section of the hippocampus. We're going to get a section of the basal ganglia. Now you can get any one of these nuclei here, either putamen, globus pallidus. Those are usually your good sections. And the reason for that is you can see a lot of motor dysfunctions. So the basal ganglia are really important for fine motor control. So for me to be able to pick up and tactfully move a scalpel, it's because I'm having a really functional basal ganglia. But some diseases can affect that, and in long-standing hypertension, we'll actually see things called lacunar infarcts. They're little tiny infarcts in the basal ganglia, and those will cause some issues. So we'll take sections of that in order to manifest, see if there's any symptoms. And then we're just going to take some sections of our cerebrum. We want to include some white matter and some gray matter, so you should be able to see the characteristic between the two different matter types. So you can just kind of grab a section from the periphery there. Now, for the rest of the brain, we'll obviously want to make sure and do our due diligence and serially section everything. So I'm going to go through and do that. Again, just be careful as you cut towards the edges there. So I'm going to cut towards the frontal lobe here. And I won't go through the whole process, but you're going to want to continue serially sectioning this whole brain. And then you can lay it out and we can look for pathologies. So just keep following that to the tips of your frontal lobe and do the same for the posterior aspect here. And we'll follow that until we get to our occipital lobe. You're going to look for infarcts, you're going to look for any sort of lesions, necrotic tissue, and as you learned in path, type of necrosis that brains undergo is called liquefactive necrosis. We don't have the same connective tissue components that we see in the rest of the body, so when parts of the brain end up dying off, they just become liquefied and get reabsorbed, so you'd actually see cavitated areas in the brain. Now I'm going to go ahead and get the rest of my sections here. So we'll get some of our cerebrum. I'll get some of my basal ganglia here. And I'll just go ahead and cut that whole square out and then just make it a little more manageable to determine any infarcts or movement disorders. And then finally, I'll get a little bit of my hippocampus. Remember, going for that little curly Q, cinnamon roll appearing structure there. Okay, so if you're keeping track, we've already gotten 4 of our 5 basic structures. Some of the things you'd be able to notice at this point too in your dissection, patients with neurodegenerative disorders will have really prominent sulci and really narrowed gyri. Particularly in Alzheimer's cases, you'll see stuff like that. And so taking samples of your cerebrum in the periphery there will actually help diagnose those particular conditions. Finally, with the brainstem removed, now we want to kind of section that and look at any potential abnormalities there. Now, you have a couple of ways you can do this. You can either do it from a superior to inferior approach, so we've already kind of cut our superior at the brainstem, and we can just go all the way to the spinal cord. Otherwise, you can do an anterior to posterior approach. I'm going to do the superior to inferior approach. That way I can see the relationship from the cerebellum to the brainstem. So again, we'll serially section, and we can lay this out. We'll look for any sort of ischemia, look for signs of infarcts, maybe potential emboli in the vessels. Notice that the cerebellum has this nice little, like, they call it like the tree of life, because it really looks very tree-like with leaves and stems and things like that. So you want to make sure that looks just like you should expect it to see here. This looks very normal. This person was in good health. And so the final section we'll take is from the cerebellum. So the cerebellum is a lot of our motor control in general. So if people demonstrate signs of ataxia, which is sort of issues with movement control, that could be a cerebellar issue. Sometimes you see that in infarcts. So we want to get a peripheral section of that, or if you see any infarcts, we'll grab sections from there as well. And so that's our 5 basic sections. So for review, we're going to look for our substantia nigra section, which should be that brown section that we saw on our brainstem, right at the midbrain component. So you should see that up there. And additionally, in the brainstem / cerebellar region, we'll get a representative section of the cerebellum. And then when we take the cross sections of our brain, our cerebrum particularly, we're going to want to get portions of the cerebrum, demonstrate any signs of neurodegenerative disorders. We'll get representation of the basal ganglia for any sort of upper motor neuron issues. And then we'll get signs of, or sections from the hippocampus to make sure there's no signs of early hypoxia for the patient. And that's your 5 basic sections. Some places, particularly if it's a neurological case, might ask for more. They might want the brain to be fixed longer, or they might have the neuropathologist actually cut it for themselves. and some places maybe even want less sections, but those are kind of a basic sections to cover a wide gamut of pathologies that may have been incidental in the patient. So that's it for brain cutting. You guys will hopefully enjoy cutting your own brains, and if you have any questions, please let me know. Thank you.